Welcome to the Salk Institute's Where Cures Begin podcast, where scientists talk about breakthrough discoveries with your hosts Ali Akmal and Brittany Fair.
I'm here today with Dmitry Lyumkis. He is an assistant professor in the Laboratory of Genetics, where he uses high-powered imaging techniques to determine the structure of tiny molecules. Understanding what these assemblies of atoms look like could reveal dysfunctions that lead to disease. Professor Lyumkis, welcome to Where Cures Begin.
Hi, Brittany. Thank you very much for having me.
Thank you for joining us today. And just kind of to start out, where are you originally from?
I am from Riga. It's the city of Riga in what is now Latvia. It used to be the Soviet Union. We moved out from Latvia about 1 month prior to the collapse. And we came to the Bay Area. I was only 6 at the time. My dad is a physicist, and like many other people from the former Soviet Union who had a background in physics and mathematics, they were heavily recruited into the United States, as well as to Western Europe and other countries as well. And so at that time in the Bay Area, there was rapid development of the high-tech industry. And my dad was one of those who was recruited.
You know, when you were coming from the Soviet Union to the Bay Area, I can imagine that was like a very dramatic change for you as a young child. Did you have any challenges when you first arrived in California?
Yeah, there was definitely a few challenges. When I arrived, I didn't know the language, and it took me a few months to learn the language. It's a little bit simpler when you're a kid, but it took me a few months to learn the language. And then what I fondly remember was the social interactions were very difficult and a little bit awkward because I didn't know how to express myself when I went into school, and kids didn't understand me. And actually, one of my best friend to this very day, he kind of guided me along that whole process and helped me out quite a bit in those first few months. To this day, we laugh about it.
That's great. And what a good friend to have, because I'm sure that was just a cultural shock. Your dad was then working in science or as an engineer in science?
He was more of an engineer. He actually was he was working in academic research in the Soviet Union very shortly prior to the collapse. A lot of those jobs started to disappear. So then when my dad moved here to the Bay Area, he started working as an engineer in industry. Okay. And that basically gave us a new life and new opportunity here.
And did you have interest in engineering at any point, or when did you really become interested in science?
I had a lot of interest when I was growing up.
Yeah.
I went through many different thoughts in terms of what I would do for a career. My real passion for science came in college.
Okay.
And it was when I actually started working in a research lab that I became really interested. Over the course of sophomore, junior, and senior year was when I really became interested in chemistry. There are thousands and thousands of different reactions and many possible ways to arrive at a result. And the idea of sort of developing your own path toward arriving at this complex molecule was really attractive to me.
I think that's really interesting because, you know, it's not every day where someone is studying something with such a passion they're not able to see.
Right. I agree. My interest began in chemistry, but soon afterwards, I became really interested in specifically protein chemistry, protein structure, and a field that we refer to as structural biology. Proteins are made up of amino acids, long chains of amino acids, and they fold into complex 3D shapes, and their function is governed by specific chemical reactions.
Okay.
For example, within the active site of the enzyme. And actually, that's a field that I continue to study and work in to this day, which continues to fascinate me.
Okay. And when you say that you're studying how proteins are assembled, like what techniques do you use to study that?
We use a technique called cryo-electron microscopy.
Cryoelectron Microscopy, also known as cryo-EM, uses frozen samples, hence the word cryo, and gentler electron beams than traditional electron microscopy to examine biological samples.
We routinely apply cryo-electron microscopy to determine the structure of proteins and protein assemblies. It's a microscopy technique that uses high-energy electrons to image objects at very high magnification. So typically atomic or close to atomic level.
Oh, wow. So you actually are able to see these tiny assemblies.
Oh, yeah. Yeah, absolutely. And the resolution can be or is approaching atomic level resolution. In fact, there was a few papers that came out, or manuscripts, preprints that came out on bioRxiv just a few months ago, which show that you can use cryo-EM techniques to image proteins at true atomic resolution.
And how does understanding the structure aid you in deciphering the function of these molecules or mechanisms by which they might be regulated?
Well, seeing is believing. A picture's worth 1,000 words. And I'll give you one example from our recent work where we used cryo-EM to visualize how specific class of small molecules interact with and bind to a protein assembly that we're interested in. We found that these small molecules bind in a completely different way than we previously expected. There wasn't a way to really predict this using any other techniques. So we had to actually get an image, a direct three-dimensional representation to really understand and have this aha moment.
So what was it like the first time that you actually used cryo-EM to visualize a structure that you were studying?
Oh, it's amazing. That's one of the coolest parts about the field, is that you can purify a protein or a protein assembly that is nanometers in size. You can purify them and you can put them onto a 3mm support grid, put it into the electron microscope, and then come out with images of these assemblies. And these images that they can lead to three-dimensional representations that are very informative.
So you came to UCSD for undergrad, went to graduate school at Scripps, and then started your academic career as a Salk Fellow. What made you want to stay on at Salk and become an assistant professor?
Salk is a very interesting place. It's a very inspiring place. Every single one of my colleagues is a leader in their own respective fields. The people here are really some of the best scientists in the world, and they inspire you on a daily basis.
Have you been able to collaborate with people outside of your field at Salk?
Yeah, Salk is one of those interesting places where all of the faculty, on the one hand, have their own individual island of research, but on the other hand, there are very deep interconnections in the ideas between different labs. And so a lot of opportunities to establish scientific connections and forge collaborations between different labs based on common interests.
And while at Salk, several of your discoveries have involved imaging tiny structures that help HIV infect cells. What really interests you about HIV, and how is imaging these structures going to help possibly with the development of a treatment?
HIV is one of the most complicated viruses that we've known about. One of the things that we've done is we've used cryo-EM to image protein assemblies that are directly responsible for establishing a permanent viral infection in a specific target cell. We described how these assemblies are put together, how they look and how they function at an atomic level. But what's important is that these assemblies are also the direct targets of one of the most important classes of drugs that are used in antiretroviral therapy to treat patients.
Were these drugs in use to treat HIV, and we just didn't know how they were fully working on a molecular level?
Yeah, so these drugs have been in use since about 2007, and some of the latest generation ones have been in use about 7 years now. They've been approved by the FDA. We are working with a team of investigators at the NIH to develop sort of third generation drugs that will be active and potent against when the virus evolves resistance. And what we found in this recent paper is that these compounds bind in a completely different way from what we expected previously. And that has important implications because in order to understand how these drugs bind and in order to make better compounds and understand how the virus evolves resistance, you really have to know at a detailed atomic level how these compounds bind and how they interact with these protein assemblies.
And since the coronavirus pandemic, have you incorporated any coronavirus research into your own work?
Yes. We have one particular project that's going on in the lab. And again, it has to do with understanding how a particular protein that is part of SARS-CoV-2 is assembled and specifically how it binds to and hijacks cellular machinery to then promote viral infection. And so what we're trying to do right now is we're trying to image this protein.
Okay. Do you think this type of work could help inform treatment options?
Yes, definitely. Understanding how viral proteins interact with host proteins can have important consequences and important implications, because if you can understand how they interact with one another, then you can potentially develop therapeutic strategies that will interfere with that interaction. And if that interaction is necessary for viral infectivity, then interfering with that interaction will in turn have therapeutic benefits.
So it sounds like in college is when you really fell in love with science. Prior to college, what did you envision doing in the future? Did you ever consider another career?
I've always liked to learn, and I've always liked the process of learning and potentially discovering. So when I started science, it was kind of a very logical career choice for me, because every single day you learn something new. You don't discover new things. Those things, that happens very rarely. But when it does happen, it's amazing. But every single day you learn something new. Science was it was a natural choice. And I've never looked back.
And what's your favorite part about being a scientist?
It's the process. And it's the fact that every single day I am learning something new. Every single day I have a little bit of a better understanding of some small part of our research or some small aspect of a problem. Once you've reached one goal, then the next goal is around the corner.
It sounds like never-ending academic growth. What is your next step?
Well, there are a number of important molecules that we would like, and important processes in the cell that we would like to understand at the molecular level. So what we're trying to do right now is we're trying to isolate and purify certain types of protein assemblies that are important, not only in virology, but also have important implications in cancer, so that we can start imaging them using cryo-EM. In the longer term, I want to see these molecules in cells. Right now, everything that we're doing is we're isolating these molecules from cells. I want to visualize them in cells, in their native environment. I think that's the next order of complexity that is just around the corner that we'd like to understand.
Thank you so much for joining us on the podcast today, Dr. Lyumkis. It was a pleasure speaking with you and learning about your work here at Salk.
So thank you very much, Brittany. Yeah, it's been a pleasure to speak with you.
Join us next time for more cutting-edge Salk science. At Salk, world-renowned scientists work together to explore big, bold ideas, from cancer to Alzheimer's, aging to climate change. Where Cures Begin is a production of the Salk Institute's Office of Communications. To learn more about the research discussed today, visit salk.edu/podcast.